the bloodline. So take a look at this.
Historically, scientists have been able to alter the traits of domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations of especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esvelt at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene-drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. Because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration and the gene drive. This same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population.
So this is a picture of the chromosomes where this light blue gene is the foreign gene that they're inserting, and the blue mosquito has it. And here's where it will cut out the gene that's normally in the mosquito and replace it with this one. And that's how this technology works. And eventually you'll end up with basically all of the offspring a couple of generations down the road expressing this foreign gene. So this is a way to sort of permanently genetically modify us. And this technology has been developed and it seems like it's ready for deployment. So